conference to the International Day of Women in Statistics and Data Science. It's my real biggest pleasure to be chairing this session and to present the work that these three researchers have done as part of the Biostatistics Unit and in connection with our group of efficient study design. So my name is Sofia Villar, and I'm a program leader at the Biostatistics Unit. I'm part of the efficient study design, which is a group of researchers that develop novel methods to increase efficiency in study design across all the stages of development and treatment evaluation. And our work focuses and spans on different areas, including those finding response adaptive designs, algorithms and devices, trial emulation, and also translation and software for implementing these designs in practice. So all the work we're going to be showcasing falls within any of these lines. So without further ado, I'll start with our session, and our first speaker will be Rajenki Das. So Rajenki is a research associate at the MRC Biostatistics Unit, and she's working on developing the statistical analysis plan and the implementation of a Bayesian adaptive randomized design for a phase 2 study called Stratosphere. Prior to working on clinical trials, she focused on modeling longitudinal mobile health data, understanding underlying digital phenotypes. Her current key interest lies in implementing and testing trial designs in real-life scenarios, accounting for different challenges. She's particularly interested in the intersection of mathematics and statistics with health, with inclination towards advancing research in mental health. So without further ado, Rajenki, the floor is yours. Thank you.
Hi, thank you, Sophia. Everyone can hear me?
I can hear you, yeah, and we can see your slides.
Okay. Thank you very much for the introduction, Sophia, and I'm so glad to be here. And a happy International Day of Women in Stats and Data Science to everyone. I'm Rajenki, and I'll be sharing what I have been working on as part of my postdoc, which is implementing a Bayesian Response Adaptive Randomization design in a rare disease setting. So what are adaptive designs? In comparison to a traditional fixed design, it's quite similar, but in an adaptive design, unlike the traditional one, we have an additional loop of conduct, review, adapt. So this gives us some flexibility where we can review our results and make some changes. These adaptations that we take during the course of the trial can be something like re-estimating the sample size or reallocating patients to the treatment groups. But one thing to note is that these possible changes are always mentioned before the trial begins and before the recruitment starts, potentially during in the protocol or in the statistical analysis plan. And then we have the Bayesian Response Adaptive Randomization, which I'll refer to as BRAR, B-R-A-R, which is again similar to adaptive design, but in this case, we update the allocation probabilities of the participants to the treatments using the Bayesian theorem. Details are in this reference, which I've provided on the slide. Rare disease. So the clinical trial that I'm working on deals with pulmonary arterial hypertension. It's a life-threatening progressive disorder, and it's classified as a rare disease. This is characterized by high blood pressure in the arteries of lungs. There's a picture provided where we can see that how the arteries can get blocked. This disease is treatable, but we don't know a cure yet, and also the exact cause is not known. The most common genetic cause of the familial PAH is mutations in BMPR2. Stratosphere 2 trial is the trial which is looking at PAH and testing 2 of the drugs. It's three-armed. That is, we have 2 treatments and 1 control arm. Phase 2 trial. So phase 2 already implies that the sample size is going to be relatively small, and then we have a rare disease, meaning it will be further smaller. Placebo controlled, which means that we are comparing the treatments to a control arm, which is placebo in this case. The participants of the study are grouped based on their mutation group, which and there are 2 mutation groups, haploinsufficiency or missense. The trial itself tests efficacy of 2 repurposed drugs. So we already do know the safety information for 2 of these drugs, hydroxychloroquine, which I'll refer to as T1, and phenylbutyrate, T2, for the treatment of PAH. And these 2 drugs target the genetic BMPR2 pathway of the disease. Therefore, the primary objective of the trial is to test the hypothesis whether the 2 treatments are able to rescue the BMPR pathway or not, and utilizes the primary. If there is a 30% increase in BMPR2, it is a success, while if not, it's considered as a failure for the treatment. This is how the trial design looks like for 1 mutation group, and it's the exact same for the other mutation group. We have 3 stages, and between 2 consecutive stages, we have the interim analysis where the allocations are updated according to a Bayesian Response Adaptive Randomization algorithm. There are 20 patients in total, divided as 6, 6, and 8 in the stages 1, 2, 3 respectively. We begin the trial with a balanced allocation where the patients are randomized to the treatments as 2, 2, 2 for control treatment 1, treatment 2. Then as we progress in the trial, for stage 2, we have the interim analysis, and depending on the how the allocations are updated or not, we again randomize patients to the new or to this possibly an updated allocation ratio. A constraint in this stage is that we do not allow any dropping of the arm. And again, as we move further to stage 3, we randomize in the similar way based on the updated allocations as per the Brar rule. And for stage 3, an active arm can be dropped. So T1 or T2, depending on what results we have, we can allow dropping of the arm, of the active arm. The Brar rule that we implement here is based on this reference by Tripa and Wason. And what it mainly does is that it protects the control arm in a way that the probability of getting allocated to the control arm does not drop under a certain level. So this is what the rule takes care of. And so given this setting of where we have a rare disease phase 2 trial and response adaptive randomization, the concern is that can we allocate the patients to the treatments efficiently, especially given the small size. So one challenge is that we want to avoid generating undesirable allocation sequences through the randomization procedure. An example description of this is that if we have randomization probability SPI, and let's say we have these allocation proportions rho, then what we would want is the expected probabilities should match the observed proportion, allocation proportion, which can be achieved in a large size trial. But when we have a small n, that is a small sample size, this becomes a concern as it gets difficult to achieve this equality or matching the 2 sizes. Therefore, the challenge is that we want to address this problem. And how we do is we propose this solution of a mapping strategy. It's an intermediate step that involves a decision rule. So in the existing trial design, we introduce this additional step of mapping, which converts the continuous probabilities, which we already derive at the interim analysis, to a target vector of discrete allocation ratios. So this is how it looks like. We have these allocation probabilities derived at interim. Then we convert these to the target allocation ratio. And then finally, once we have the allocation ratio for the next stage of the trial, we generate a random sequence based on this discrete ratio. We propose two types of mapping. One of lower granularity, which has lower number of thresholds, and the higher granularity one has higher number of thresholds. To achieve this discretization, we use some thresholds, which I'll be talking later. And this is the adaptation list for this trial. That is what kinds of adaptations are possible within the trial. And these columns represent, for the first one, its adaptation category. The kind of adaptation description is just describing what the adaptation means. And for the last three columns, we show whether the specific adaptation category is an option for that stage or not, because we already had some constraints in place. For example, starting with drop, if drop means that T1 or an active arm can be dropped. Here for this representation, I have only taken T1, and as you can see that for stage 3, dropping is possible, and then we may have an allocation ratio of 2:0:6. Had it been for T2, then the ratio would have been 2:6:0. But here, for simplicity, we are keeping T1. Then we have disfavor, which means, according to our design, that an active arm, here T1, can be disfavored but not dropped. So again, in stage 1, that's not possible. Stage 2, that is possible, and it may result in 2:1:3 allocation ratio. Or for stage 3, we may have two of these ratios, and we pick one of these randomly if we need to disfavor. Then we have balance, which is an option for all the three stages, and then favor and keep, which follow a similar meaning as disfavor and drop, but in the opposite way. Like I said, to achieve the discretization, we are implementing the use of some thresholds based on which we name the two mapping strategies. Here is a schematic of the proposed higher granularity design. And for stage 2, you can see that we don't have the options of drop and keep, which I again specified in the last table. And for stage 3, we have again 5 different categories. And there are these thresholds in place which decide that depending on where the allocation probability falls, the category is chosen. And to revert to the lower granularity, all we do is close the balance region. So for example, for the first stage, stage 2, we simply equate the two thresholds. That is, both thresholds are equal, and the only decision that we have to make is between disfavor and favor. Similarly, for stage 3, once we close the balance region, we are left with 4 categories on the decision line. Here is the summary of all the response adaptive randomized designs that we consider in order to compare our map design with rest of the designs, whether it's performing well or not. we have 3 unmapped design where we do not have this mapping rule. And starting with fully unrestricted, there is no constraint in any of the stages, and we implement a very basic Thompson sampling. There is no restriction on number of control arms. For control protected, we do not have any restriction on the 3 stages. And then we implement the 3-par rule, which I had mentioned before, but it's basically a refined version of Thompson sampling, which protects the control arm with the help of tuning parameters. Stratosphere 2 is the one that we consider, and this is where we implement our mapping. For Stratosphere 2, we have all the constraints in place. For the mapped, again, we have all the constraints along with the number of control arms fixed as 2, which is aligned with the 3-par rule of protecting the control arm. Given those designs, here is how the operating characteristics, where the significance level is set to an alpha of 12.8% to meet 10% error control. Here we see the last 3 are what we focus on. And we do see that the power improves and even the patient benefits improve as there's more allocation to the superior arm. This is an example of how the first interim can look like. So we have the number of successes, which is based on the primary endpoint, which was the change in BMPR2. And then we have the allocation probabilities utilizing the Brar formula 3-par rule in this context. Then we have these mapping steps. So we decide whether I have not disclosed the value of threshold because the trial is still ongoing. And then finally, based on what decision we make and how the thresholds look like, we decide the target allocation ratio. So during the interim, our question is, do we adapt the randomization ratio for stage 2? In this example, yes, we change from balanced to favoring the T2 arm, in this case, phenylbutyrate. To conclude, this proposed mapping criteria ensures good performance of the randomization procedure. It provides a viable strategy that can empower especially small size trials while we still stay true to the essence of RAR, response adaptive randomization. This additionally also makes the implementation of RAR easier through external randomization providers like sealed envelope, where we are often asked to input just the final randomization sequence, and in that case, discretization is really helpful. And one of the possible extension is incorporating different trial designs with mapping in place, which can be a larger sample size or more number of blocks or bigger sized blocks and so on. These are the references. The highlighted one explains the mapping criteria, and it was submitted in arXiv recently. Hope this is useful. Thank you very much. Any questions, comments?
Thank you, Rajenki, for the very nicely presented talk. There are no questions at the moment, but we can save questions for the end. I have just one quick question while we let Mia get ready with her presentation. Can you comment a little bit on the level of blinding of Stratosphere and how this was factored into the design?
Yes, so thank you. I'll stop sharing maybe. It's called a veiled blinding. What we do is compare the 2 treatments with the control arm. So by veiled, so people might have heard of a double dummy approach, where if we have 2 treatments which are of different types and noticeable changes like physical appearance or way of taking the medicines, then one may usually allocate 2 different controls so that it's completely blinded. But in this case, a patient gets only one kind of pill, which can be T1, T2, or C1, corresponding to the particular T1 and or C2. So it's veiled because the patient still does not know whether whether they are getting a placebo or an active treatment, but they can guess that it's not the other kind of treatment. So it's the veiled.
Thank you, Rajenki.
Thank you so much. Yeah.
So our next speaker of the session is Mia Tackney. So Mia is a research associate here at the Biostatistics Unit. Her research focuses on statistical methodology for using digital devices to measure outcomes in trials. And she has particular interest in how to deal with missing data and developing missing data methods. She will be presenting our recent paper, 8 Methodological Questions for Digital Outcome Measures. And this is joint work with James Carpenter. So Mia, the floor is yours.
All right, thank you very much. Thank you for inviting me to be part of this session. Before getting started, I wanted to acknowledge all the lovely female statisticians that I've been very fortunate to have as supervisors and mentors. So they include Rosemary Bailey from the University of St. Andrews, Elizabeth Williamson, or Fizz, with whom I worked at the London School of Hygiene and Tropical Medicine, and Sophia Villar, who I work with now at the MRC Biostatistics Unit and is chairing this session. So in this talk today, I'll be discussing this paper, which is about methodological challenges when it comes to using digital outcome measures. And this is joint work with Sophia Villar and James Carpenter. So to begin with, in clinical trials, we measure health outcomes of patients in order to assess whether a new intervention is safe and effective. And in many disease areas, these health outcomes are measured in a clinic setting. And in one example that I'll be talking about, this is a rare disease. We already heard about rare diseases from Rajenki, but one example I'll be talking about is from Duchenne muscular dystrophy. This is a condition that affects young boys and often leads to progressive muscle weakness. Here, the gold standard endpoint is a 6-minute walk test, which requires patients to come into the hospital. So it can be a really long journey for people who are affected by these rare conditions. And the outcome can be affected very much by how tired or how motivated the child is. This is really a kind of snapshot measure. And with the use of digital technology, there's opportunities to make things more convenient for patients, to allow patients to have their health outcomes assessed in their own home. But if you want to use such a novel health outcome as a primary endpoint in a trial, you need regulatory approval for this. And it was considered a landmark event when last summer, the European Medicines Agency approved for the first time a digital endpoint as a primary outcome measure in Duchenne muscular dystrophy. So when you use a digital outcome measure, the hope is that you get a lot more data. These accelerometers, in this case, it's worn on the ankle, and it measures acceleration in 3 dimensions, 100 times per second. So it's a lot of data. Here I'm plotting just 30 minutes of what that data looks like, because that's about as much as you can plot before your computer crashes, because it's just so heavy. And you can collect data over a longer period of time. Here I'm showing what 1-minute epochs look like over a week. And you can hopefully summarize this in a simple way, and the summary will be somehow better than what you get in a in-person 6-minute walk test, you hope. But through this process, there's lots of statistical challenges and lots of new questions, lots of new opportunities, but also areas of discretion where there's not much guidance. So what we wanted to do was to highlight these potentially very new questions that don't yet have answers. And some of those questions are around the validation of these digital outcome measures. So around this question of how do we know that the digital approach is just as good as the traditional approach? And some of the questions are about embedding these digital outcome measures into trials. How do they change the design and analysis of trials that use them? So I'll go into these 8 questions, but before that, I first want to give an overview of what validation for digital outcome measures looks like. At the moment, there's a very recent framework that was proposed by the Digital Medicine Society called the V3+ framework, and it breaks down validation into these 4 different components. The first is verification. So this is all about the hardware, the device, making sure that these work sufficiently well via benchtop tests in the lab. The second component is about usability. So this is about whether patients can accept and easily use this approach. Next, analytical validation. This is about the physiological metric. So for example, if you want to measure the strides, as we want to do with stride velocity 95th centile, you want to make sure that those strides that you measure with the accelerometer are just as well measured as you would be able to do in a motion capture room. They should also be repeatable. So if you measure stride velocity 95th centile at one period of time, and soon after you measure this again, they should be quite similar. And you can assess this with the intra-cluster correlation. You also want an assessment of the robustness of this measure to understand what is the impact of changing the measurement period and impact of contextual factors, such as if it's a weekend or a weekday. We then come to clinical validation, and this is about being able to identify a clinical state. So for example, they showed that for the stride velocity 95th centile, the patients who had Duchenne muscular dystrophy, their values were lower than their age-matched controls, as you can see by comparing the red dots and the blue dots. They also showed that the stride velocity 95th centile was highly correlated with the traditional approach, the 6-minute walk test, as you can see by the gradient of this plot. And finally, you have to show that the new digital approach is sensitive to change, both in terms of disease progression and treatment. And in fact, for stride velocity 95th centile, they showed that it was more sensitive to change. And because of this, you could potentially have clinical trials that require fewer participants, simply because of the sensitivity of the new digital outcome measure. Now, when it comes to this aspect of validation, we wanted to ask several methodological questions. And the first is, what is the appropriate outcome granularity? So as we saw, some aspects of validation require data at a different level of granularity than others. For example, if you want to assess whether the strides are accurately measured, this requires data at a different level compared to if you're looking at whether the summary metric, the 95th centile of strides, can appropriately discriminate between people with and without the disease. But as you move through these different levels of granularity of data, lots of decisions have to be made, for example, on what the epoch level should be, what you should do with outliers. You may not even have access to every level of this data, depending on what kind of device you're using, what kind of software you're using. So there's a lot of areas where there's not much guidance. The second question is, what is the appropriate measurement period? So patients will have to use this device, interact with the device for some amount of time. And the longer it is, the less variable the summary metric will be. But you do have to consider things like the compliance, how much burden it is for the patients, and also aspects like the battery life. Thirdly, there's the question of how you summarize all that data into one summary metric. And in the case with Duchenne muscular dystrophy, they chose the 95th centile because it was more sensitive to change than other metrics that they considered, such as the median of the stride velocity. But it's not very clear how you should make this decision in other contexts, especially because there are so many criteria around validation that you want to fulfill. And lastly, we know that when you ask participants to wear these devices in their daily conditions, there's likely to be data that is missing. It could be quite complex, the types of missingness that you find. There may be entire days that are missing or parts of days that are missing. And it's very likely that it will be missing not at random. So there's a big question around how to define and handle missing data. So we now come to the questions around embedding these digital outcome measures in trials. There is some guidance on this, for example, this document by the FDA. However, there is not a lot of guidance on the statistical aspect. So that's what we'd like to highlight in this next set of questions. So in this next question, while we acknowledge that a primary outcome, primary analysis is likely to look at the summary metric, for example, the stride velocity 95th centile, you could take advantage of the high-frequency granular data in order to learn more about the treatment. So, for example, this may help you to understand about changes in the physical activity during the day. In this paper by Lizzie and Abellan from GSK, they showed that in patients who have ALS, as their disease progresses, their physical activity declines in general, but in fact, you can see that there is a change in the middle of the day, and that's something you would not be able to appreciate if you relied solely on analyses that take some kind of summary metric. There's also potential to learn about the early signs of efficacy and even about the mechanism of action of treatments. But in order to answer these kinds of questions, you may need to use more flexible modeling approaches, for example, generalized additive models, which they use in this paper here, or perhaps functional data analysis or even time series analyses. So these are methods that are typically not used in the clinical trial space, but may be very relevant as digital outcome measures become more prevalent. Related to this is the question about seasonal or behavioral factors that need to be accounted for. So when you measure people's physical activity in their daily conditions, you are no longer measuring exercise capacity, but also their preference towards exercise. And we know that that tends to increase in the summer and decrease in the winter. So if you imagine someone who is recruited at baseline in winter, and at follow-up, they have their assessment in the summer, you can see that it's possible that this difference will be impacted not only by the treatment, but also by a seasonal effect. Now, thanks to randomization, in general, this will not incur a bias. However, it can lead to increases in standard error. You may have similar considerations for observer and learner effects. So these are all things that you need to have in mind when you're thinking about the recruitment of the study, the randomization, and how to do the analysis, whether you need to adjust for these factors. The next question is about what are appropriate estimates for digital outcome measures. When you change from a traditional to a digital outcome measure, this can impact each of the attributes of the estimate. So for example, it's possible that if you use a digital outcome measure, your targeted population or the people that you're able to recruit in your trial may be more able to reach those who are in geographically remote locations, but you may exclude people who are unwilling or unable to use digital devices. Secondly, if you use a digital outcome measure that is able to measure people's daily activities, this may capture the scientific question better if it is about patients' experiences and their physical activity in daily life. Next, using a digital outcome measure may change the type or frequency of intercurrent events, especially those that are related to technical issues with the device. And as we have already discussed, it can change how you derive the population level summary. It may be more complex when you have a digital outcome measure. In this paper by Isam et al., they describe how decentralization, not just digital outcome measures, but decentralized trials can impact each of these estimate attributes. And they invite people to think strategically about these decisions when they're thinking about decentralized components. Our last question is whether evidence synthesis is possible. So we're seeing that there is a vast heterogeneity of digital outcome measures, particularly as exploratory endpoints. In a recent review, they found that there were 266 digital outcomes from 21 diabetes trials. So these are different summaries that you can get from digital outcomes and doesn't even go into the different decisions that you could make, for example, about the measurement period. So going forward, there's a big question about whether we can do evidence synthesis. So this is a summary of the 8 questions which we present in this paper. We also include a simulation study which highlights the impact of these questions in a trial that's based on a recent Bellerophon study, which uses time spent in moderate to vigorous physical activity as a digital outcome measure. So thank you very much for this opportunity and for listening. Yeah, that's it for me.
Thank you, Mia, for the very exciting presentation and lots of work to be done for answering all of these questions. While I let Nina set up for her talk, I'll make one quick question. Can you tell us a bit more about learner effects and observer effects? And also, are there examples of seasonal trends that are seen in trials anywhere that you're aware of?
Yes, thank you for this question. So observer effects, they typically lead to a slightly change in behavior in participants when they know that they're being observed. So for example, when participants first receive the wearable device, they may be more active because they are aware of being observed at that time. Learner effects may be more relevant for digital outcome measures that require some kind of activity, some kind of active task. So this could be a digital walk test, or it could be entering patient-reported outcomes into an app. This may all have learning effects. In terms of a seasonal effect, something we explored was in our paper that was based on a Bellerophon trial. Participants were recruited over a time period, but it's not known to us from reading the literature where during the seasons the patients were recruited. So we explored a few different possibilities for how that could have happened and how that may impact the treatment effect estimates. So yeah, that's something that you can look more into in the simulation study.
Thank you. We'll move on to our next speaker. Thank you, Mia. Thank you, Rajenki. Our next speaker is Nina Deliu. Nina is a researcher in statistics in Imhotep Sapienza University of Rome. She's also a visiting researcher at the MRC Biostatistics Unit in the University of Cambridge. She is part of the editorial board of Young Stats, the blog of young statisticians in Europe, and has curated the Women's Statistics and Data Science Twitter account. Nina's primary interest is devoted to sequential decision problems intersecting areas of inference Bayesian statistics, multi-arm bandits, and the design of experiments. Her research is inspired by challenges arising in real life, and she strongly believes that the methodological progress should go along with the concrete real-world needs, and to be not to simply be good, but also good for something, which is a really good motto to have. It's my pleasure to give the floor to Nina. And Nina, looking forward to hearing your presentation.
Yes, thank you very much, Sophia. Thank you for the
We can't hear you.
Oh, okay.
Sorry, try again.
Yeah, can you hear me now?
Yes, yeah, now we can hear you. Perfect.
Okay.
The slides are there. You can start now.
Right. Okay. Thank you very much. And sorry for this small interruption. So thank you very much, Sophia, for organizing this session and for inviting me to share some work I've been doing during my postdoc at the unit. So the work I'm going to talk about relates to some results on a novel test specifically developed for response adaptive randomization designs. And these results refer to some asymptotic but also फ़inite-sample properties of the test. So to start with, as you mentioned at the introduction, my main interest is devoted to sequential decision making, and you can approach this interest in 2 ways. From one way, you can approach it as an offline learning or estimation process where you already have the data, and what you aim to do is to learn from those data and estimate some policy. For example, you do this in dynamic treatment regimes. On the other side, some other times you may want to learn online as new data accumulate. So for example, you start with no data and then you observe the data you collect, what you allocate, so the arms, and the responses to those arms, and you want to optimize this kind of design. So this is typically used a setting for sequential design of experiments. And this is the context I will be focused on in this talk. And more specifically, the guiding questions that I've been focused on during my postdoc at the biostats units related to this context are how can we design efficient, safe, and theoretically principled algorithms for adaptive experiments that can solve practical problems in areas such as clinical trial, mobile health that may use digital endpoints as the one mentioned by Mia, or other fields such as education. And on the other side, once we have collected this data, how can we perform valid inference? So adaptively collected data can be, for example, data collected through some sort of response adaptive randomization. And response adaptive randomization, it's good and has been advocated as something to pursue in clinical trials. Why? Because what you will do, you will have a set of participants, say N, you will allocate some arms to these participants or treatments according to some policy, to some rule. And then once you observe the outcomes associated with the arms that you have allocated, you may want to update this policy. You may want to update it in order to reach, for example, some desiderata for your trial. One of these is to maximize the cumulative responses over time, which may be also equivalent to minimize the exposure to inferior arms. So if you realize that during the trial there is one arm which is less promising, you may want to decrease the allocation of this arm or just to drop it, as Rajenki, for example, mentioned at her first talk. But since you are running an experiment, you also want to maximize these responses. So you want to earn, but also enable inference. So you also want to learn about the treatments that are being allocated. So here comes some challenges, because you have two goals. And if you want to pursue the two goals in parallel, you may have some compromise. So you want to understand what is the trade-off between these two. So let me show you some challenges that you may incur during this kind of trials. So let's assume a very, very simple trial or experiment in which you have basically 3 stages. In these 3 stages, you allocate 150 participants to 2 arms. And you simulate some data according to a normal distribution where the 2 arms are basically equivalent. So they have the same mean and they also have the same distribution. So let's suppose that we adopt this kind of response adaptive randomization design, which is very simple. So at the first stage, we allocate arms with an equal balanced design. At the end of the first stage, when we have our 50 participants, we look into the arm associated with the highest mean. And in the subsequent stage, we allocated 80% of times. And we perform the same at the end of the second stage. So if you look at the distribution of our estimator, and our estimator here is the mean associated with one of the 2 arms, what we can realize is that at the end of the first stage, in which data were allocated in a balanced way, we have a nice normal shape and there is no bias. But as soon as we move from this balanced design, we can see that this estimator, if used throughout the trial, it introduces some bias, and we also kind of diverge from the expected normal asymptotic distribution. And this challenge can get even worse if we use more complicated design. So the design I'm mentioning here is the Thompson sampling, is a Bayesian response adaptive design, is a kind of preliminary version of the design presented by Rajenki in her first talk. And the idea here is that we want to allocate the arms, for example, arm K, according to its posterior distribution of leading to a mean which is higher or equal to the mean associated with all the other arms. So basically it's based on a posterior update in which we update the mean distribution of each of these arms, and we allocate the arm according to their posterior expected mean. Why this design? Well, there are a lot of reasons of why using this design. Two of these are that it presents an optimal solution for the so-called regret minimization bandits, which I'm not going to touch here, but basically the idea is that you want to minimize some regret or maximize some reward, and it has a very easy iterative implementation if you have conjugate families, for example, the normal one and others. Another reason is that it's perhaps one of the most commonly used response adaptive randomization designs outside trials. So it's something interesting to explore. So for example, we use this design to conduct a real-world experiment through a platform, Amazon Mechanical Turk, in which we basically send two types of messages and ask participants to rate the two messages in terms of how helpful they think these messages will be for helping them to manage their stress or mood. And what we realized at the end of the experiment, over 150 participants, is that the allocation probability very quickly skewed towards one of the two arms. And this is what you can see with this red line, which basically reports the allocation probability of arm 1. And at the end of the trial, 137 participants were allocated to this arm. So of course, if it's true that arm 1 is a most promising arm compared to the other one, you are performing some earning. Okay, so the ethical goal of a trial is achieved, but in the end, if you will have to learn, so for example, to test hypotheses from this data, where you have 137 participants allocated to arm 1 and only 13 allocated to arm 0, this may be very challenging with standard statistical tools or standard statistical testing approaches. So there are some solutions in the literature. Maybe now you will find even more than the ones that I've been comparing our proposal here. But the proposed valid solutions are typically limited to large samples because they are asymptotic solutions. They have reduced power. So again, if you have small trials, they are not suitable, and they require some restrictions on the design. So for example, you will not be able to use the standard Thompson sampling. So we asked, can we do somehow better? In particular, can we preserve point number 3, in which we still optimize the trial in the sense that we preserve design's optimalities, but move away from the earning-learning dilemma, so still improving in our inference. So okay, so what we the short answer here is we did our best to do that. So perhaps some solutions may exist to move away from this dilemma. So it's not always a compromise. So let me explore these solutions. So basically, in 1985, there was a quotation by Peter Armitage. So in one of his works, he said that the allocation proportions are moving away from 50-50 balanced design because the evidence points to the superiority of one treatment. So why not using the evidence given by the design itself? So for example, if for one of the two if under an alternative hypothesis, so if what you observe is an allocation probability that moves away from the balanced design towards either 1 or 0, then perhaps it happens for you to be, say, on average, most likely in an alternative case where one of your arms is indeed a better arm, a most promising arm. So again, based on inspired of some recent work, which is quoted here, published in Biometrics, we basically looked into this allocation probability test. And this test is basically framed as a function of the allocation probabilities directly. So in a two-arm setting, in which we have arm 1, arm 0, say, where 0 is the control arm, we look into the indicator function of our allocation probability being greater than some threshold. For example, here we took the 0.5 because it's the probability that you will have in a balanced design. So we look into how many times this allocation probability moved away from a balanced case in which it should have been 0.5. So we try to study the properties of this test defined in this way. So to illustrate some of the properties of this test before going into its theoretical, so let's look into some examples. This test is a discrete test. It is the number of, it is a number, a discrete number, and it has this kind of shape. Under the null hypothesis, in which you don't have any best arm, any optimal arm, it looks like a symmetrical distribution. And given the critical value, or where the critical value of a test is basically the value which ensures that you have some guarantees on the type 1 error, alpha here, you can also interpret this distribution, more specifically the right bars of this distribution, as your type 1 error or power. So perhaps this is more easily seen in the alternative case, where the right bar is indeed the power of your test. So here, given the same critical value developed under the null distribution, we get something which is not symmetric anymore, is something which is skewed to the right, and is skewed to the right because we have arm number 1, which basically drives this test, drives the allocation probabilities, which is by definition, by hypothesis, the optimal arm. So you see that this kind of test has very different shapes from the one that you will have in a standard statistical testing approach based on asymptotic distributions. And at the same time, if you were to compare the properties of such a test statistics based on its distribution with something based on asymptotics, you will gain much more. So for example, based on a valid test introduced in the field of response adaptive randomization designs, we are able to improve in terms of type 1 error and at the same time to gain in terms of power. So with novel testing approaches, you kind of move away from competing goals, competing interests between learning, but also between type 1 error and power. So let me see how much time I still have. Okay, so let me move very quickly towards some of the distribution of this test. Since it is a discrete distribution, by working with some math, we were able to compute the final sample distribution of the test under the null setting. This is what we want because our critical value is derived under the null. It has this shape, okay? So I know it's quite complicated while looking here, but what is enough to see here is that it depends on some correlation structure between the allocation at the different time points. And it also depends on this PT, where PT is basically the probability of observing an arm, observing responses for that arm improved in comparison to the other arm. So these are the main quantities on which these statistic distribution are derived. Second, in terms of some asymptotics, we can see that the test diverges under the alternative. And in particular, it diverges under the alternative and a Thompson sampling design. In general, it will diverge if this probability here will, its limiting value is going to be some constant. some constant greater than 0.5 or in general greater than 0. And this happens for Thompson sampling because since it is an optimal design, this allocation probability, if the arm 1 is an optimal arm, converges to 1, converges to allocating that arm with probability 1 in the limit. So it diverges under Thompson sampling. And why is this an interesting property? Because since this divergence is much more slower under the alternative, you are ensured that you will have basically good power properties. Other theoretical results related in this case to the design under study, which is Thompson sampling, that in addition to computing the distribution of the test under the null for deriving its critical value, under Thompson sampling and other Gaussian setting, we are able to derive the distribution of the test also under the alternative. So we are able to compute exactly the power of this test under, of course, a specific setting and a specific design. So let me now conclude with our experiment, the experiment that I've mentioned you before. So in this case, as mentioned, having two arms with such a big imbalance will basically allow you no statistical inference and especially no power if you were to apply some standard inferential procedure. But in the case, if we move away from standard procedures and we try this new approach based on the allocation probabilities, we have validity of the procedure first. And also in this case, we are able to actually reject our null hypothesis. So to see this, perhaps the most easiest way is to perform some simulation studies. So to try to understand the properties of the test under a design perspective, where you basically run the same trial with all the different inferential procedures. So here we have compared other two existing approaches in the literature, two asymptotic approaches. And what you can notice here is that fixing the type 1 error to a 0.05 value, with the proposed AP test, we are able to ensure a smaller regret, so a higher cumulative number of responses, because we preserve the design's optimality, while at the same time improving in terms of power. So, for example, with an alternative test, a batched OLS, so an OLS variation, we are performing very poorly in terms of power because asymptotics are based on having very big batch size. So asymptotics are based on the size of each block in which you adapt your design. So overall, with the proposed test, we basically gain in terms of all of the three parameters here: type 1 error, power, and regret. Sorry, not all of the three. Type 1 error is fixed. So we gain in power and we gain in regret. So in conclusion, since in many experiments you are tight in terms of the size of the trial, for example, you may work with pilot trial, and still you may want to pick up the signal quite early. You don't want to lose earning. Using a finite sample test in which you have validity in finite samples is a very good alternative, because you can at the same time improve in terms of robustness in different settings. You can improve in terms of power. So while earning, you can still show and perform some inference at the end of your experiment. So, and overall, it represents a flexible approach, flexible and interpretable approach. So thank you very much. Hope I am on time. And if you have any questions, I'm happy to ask. Okay.
Thank you so much, Nina. Thank you very much. Before we hand over to the next session, can I make a very quick question? So you talked about this T-min, and in most RAR, we have this burn-in that is not to do with the testing. So can you clarify what you think? Is that the same or can it be different?
Yes. So T-mean here mainly refers to, say, a minimum number of time points, where the time points for this test are intended in the sense that how many times you have adapted your allocation probabilities. So let's say that you have a trial in which you have 3 stages, such as the one introduced by Rajenki. In that case, you have 3 time points. In that case, if your first stage has recruited, say, I don't know, 50 patients, it may be enough for you to have gained enough information in order to start looking into the allocation probability at the end of that first stage as something which is guided by the evidence. So this T-mean will reflect the size of each stage that you have in your trial. If you have a fully adaptive trial in which you adapt at every new patient, you may want to move farther away from 1. It's something maybe we should work a bit more on, at least with simulations, but intuitively, this is the idea. So it's the minimum, it reflects a kind of minimum sample size after which you are happy to use the allocation probability as something that is based on the evidence and is not just noise driven.
Thank you. Thank you very much. So I'll thank our three brilliant speakers, Mia, Rajenki, and Nina. And thank you for agreeing to this invitation and for the wonderful session. And we can make a snapshot of all of us while we're here. And we can certainly hand over to our next colleagues that are running the following session. Thank you again.